What if I told you that you could directly manipulate a genome? I'm Caitlin, and on this week's episode of What Makes the Cut, I'll show you how you can do just that using the CRISPR-Cas9 system, where CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Here's basically how it works. Imagine you're writing a college essay. It's the due date, so it's crunch time. You're almost ready to turn it in, but you have to proofread it. As you're going through the last sentence, which you wanted to say, and this is why I'm on Santa's nice list, you find that your 2 a.m. self has written, and this is why I'm on Santa's naughty list. Uh-oh. So how do you fix it? You identify the error, delete it, and replace it with the correct word to give the most important sentence of your essay the correct meaning. You're indeed on the nice list. Okay, so how does this very broad example apply to a biological system? In our example, if our sentence represents DNA, the nice naughty error represents incorrect DNA, and then CRISPR acts as the editing tool used to correct and manipulate it. This function of CRISPR and its associated enzyme, Cas9, was first found as an immune response in bacteria, where foreign DNA was recognized as harmful. Cas9 acts as biological scissors to identify, cut, and degrade this DNA. Once this was discovered in bacteria, we thought, why don't we see if this system works on other organisms? A modified Cas9 enzyme has made this possible. For a very general example, say you want to knock out the gene controlling orange scale color in goldfish. Although many genes may be involved in this, for our purpose, we will assume there's only one. As a scientist here, we need 3 things for Cas9 to work in our goldfish. First, we need a target sequence that is complementary to the color encoding gene. Second, we need our target sequence to be right next to a PAM site, where PAM stands for Protospacer Adjacent Motif. This is a 3-nucleotide sequence that Cas9 will recognize to make its cut. Third, we need a guide RNA, which will guide Cas9 to the right gene, in our case, the scale color-causing gene. These sequences will be incorporated into a plasmid, where they can be injected into goldfish embryos. Once operating in the fish, the process will look something like this. The guide RNA will call Cas9 to create a guide RNA Cas9 target site complex. The target site will find the location in the fish genome to which it is complementary. This complementation between target sequence and gene will cause the target site and Cas9 complex to bind to the fish's gene. Once bound, Cas9 will recognize the PAM site and cleave the DNA. The break in DNA will be enough to disrupt the gene. If the gene is properly disrupted, it cannot produce the orange color-causing protein. This means that our engineered goldfish will be colorless. In our example, we caused a knockout of a gene. However, in more involved processes, Cas9 has also been used to activate, repress, or replace DNA sequences. As scientists, we're always looking for solutions, so this leads us to think, what if we could use CRISPR in humans? On cancer-causing genes? What about in replacing defective genes that cause lifelong effects? The implications of this genome editing tool on human health are monumental and remind us that humanity and science are not separate, but rather one and the same.